Welcome to FYI, the For-Your Innovation podcast. This show offers an intellectual discussion on technologically enabled disruption, because investing in innovation starts with understanding it. To learn more, visit arc-invest.com.
ARK Invest is a registered investment advisor focused on investing in disruptive innovation. This podcast is for informational purposes only and should not be relied upon as a basis for investment decisions. It does not constitute, either explicitly or implicitly, any provision of services or products by ARK. All statements made regarding companies or securities are strictly beliefs and points of view held by ARK or podcast guests and are not endorsements or recommendations by ARK to buy, sell, or hold any security. Clients of ARK Investment Management may maintain positions in the securities discussed in this podcast.
Hey everyone, welcome back to FYI, ARK's For Your Innovation podcast, where we talk about disruptive innovation and interview lots of interesting people. Today I'm here with Ali Ehrman, my fellow analyst. I'm Tasha Keeney. Ali, what are we talking about today?
We're talking about what I always love to talk about, which is gene therapy, gene editing, sort of where it came from, what it evolved to, base editing, prime editing, proteome editing. I mean, there's just so much we can talk to. Our guest today is pretty amazing. So I'm excited. I'm excited to get into the conversation.
Okay, so editing of all kinds. And why do we care so much about gene editing? Why are we always talking about it?
Yeah, so, first of all, it could potentially cure disease, which could be huge. We've seen some really interesting data come out, from CRISPR Therapeutics and others, that sickle cell anemia and beta thalassemia could potentially be cured by a gene edit, which is really, really exciting. We're also seeing that gene therapy and editing trials are basically increasing fivefold since 2010. So today we have about 10 approved drugs. We have many in clinic. And then if you think about that, sort of adjusting for the typical trial failure rate, we're thinking about 170 gene therapies could be approved and commercialized during, the next decade. So a lot coming down the pipe to be excited about.
Wow. Okay. So that's, you said 10 today, over 100 within the next decade. So today you spoke with David Liu. Tell me more about this guest.
Okay, so first of all, I would say that he is one of the most prolific inventors and scientists of our generation for sure, maybe ever. One of my personal heroes. I mean, you can't even describe this guy in 2 minutes. So I'm just going to do the bare minimum and encourage everyone to look him up online. He does a great tour of his lab in a video. So encourage everyone to check it out and just see how cool and innovative he is, even within his own lab. But just super briefly, he's vice chair of the faculty at the Broad Institute of Harvard and MIT. He co-founded many companies like Prime Medicine, Beam Therapeutics, Editas. He also went to Harvard and graduated first in his class. I mean, he's published, hundreds of academic papers. He's an inventor on, over 70 issued U.S. patents. So not enough time to discuss just how cool this guy is and how much he's accomplished in a very short time.
Okay, well, yeah, it sounds like a very accomplished guest. I'm excited to hear what he's going to talk about. What do you think are, the most exciting things that'll come out of today's discussion?
Yeah, I mean, I'm really interested in base editing and to see where it can go. I think sort of the multiplex editing ex vivo, so doing a lot of edits outside of the body is just a really interesting concept. And base editing has made that a little bit more simple. So, excited to dig into that and to dig into proteome editing or editing of proteins. I think just understanding from the person who's actually, discovered some of these things in the lab, but is still able to communicate it in a really digestible manner will be really great for me and for our listeners.
All right. Well, I know I'll certainly learn a lot today. I'm excited to hear it. So let's get right into it.
Hello, welcome to FYI For Your Innovation podcast. Today is an exceptionally exciting day for me because I get to interview one of my personal heroes, Dr. David Liu. So thank you so much for being on the podcast and welcome.
It's a pleasure. I'm really excited to get the chance to chat with you.
Cool. So firstly, it looks like you're in space. So can you tell us a little bit about where you are?
So this is a room in our home above the garage, and I really love projects that marry science and art. My house is full of do-it-yourself science plus art projects. So this was a simple one where the sky is a fiber optic sky, illuminated with LEDs that come from a vegetable colander that I converted into an LED illuminator. And it's actually the actual Boston summer sky. So you can see Orion. People might be able to recognize Orion's belt is the three dots in the middle of the screen. And you can see Gemini, and the Dipper is over here somewhere. And so it's just a nice tribute that sometimes in Boston you have to be indoors because it's a little bit cold. But if the walls, it's oriented the right way also. So if the walls were to fall in June and you look up, that's what you would see.
It's amazing. That's really cool. So let's start off easy here. In your TED Talk in 2019, which was pretty incredible to listen to for me, I thought you did a really, really great job of explaining just what base editing was, maybe some advantages over other forms of editing. So would love to just maybe start there.
Yeah, sure. So base editing is a strategy for changing the genome that builds on the seminal work that Jennifer Doudna and Emmanuel Charpentier and Fun Zhan and George Church and Virgis Siksness and many others contributed to in discovering and developing the ways that CRISPR agents can sequence specifically bind to DNA. And of course, the work of many researchers before them in using zinc fingers and tail arrays to also bind DNA in a sequence programmable manner. But with base editors, instead of binding or cutting the DNA, which is what CRISPR-Cas9, zinc finger nucleases, and TALENs all do, instead of cutting the DNA double helix, cutting the chromosome into two pieces, base editors instead perform chemistry on an individual DNA base at the target site in a living cell and convert one base to another base. So base editors actually rearrange the atoms in one DNA base, like C, to become instead a base that looks like T. Or they rearrange the atoms in an A to instead become a base that pairs like G. And so by directly changing one DNA base to another base, you can change the coding of that gene without perturbing the cell and the chromosomes the way that causing a double-stranded break tends to do. And so that has certain advantages and certain strengths compared to strategies, more traditional strategies of cutting the DNA double helix.
So would one of those advantages be off-target edits?
Yeah, I think there are a variety of different strengths of various gene editing platforms. One of the strengths of a base editing platform is we can make specific changes from one sequence to another sequence without generating a mixture of uncontrolled products, byproducts. And what many people may not realize who aren't in the field of gene editing is when you use molecular scissors like CRISPR-Cas9 to cut a DNA double helix, to cut a chromosome into two pieces, the cell's reaction in most cases is to try to get those ends back together, but that process can occasionally make mistakes. If it doesn't make mistakes, you end up with the starting sequence that you began with, and then the scissors can cut it again. So eventually you get to products that have these mistakes, these insertions and deletions of different numbers of DNA letters. But the thing is, we can't control what those indel, as they're called, those indel byproducts are. So for that reason, when you cut DNA, when you use CRISPR-Cas9, most of the applications, and right now all of the 43 clinical trials using CRISPR, they're all using them in a way to disrupt genes. Because making these uncontrolled insertions and deletions at a target site is a great way to disrupt a gene. But for most human genetic diseases, you need to precisely fix a gene in order to have anticipated therapeutic benefit, as opposed to disrupting the gene. Now, for some diseases, there are very clever ways you can disrupt a gene to help a patient. Sickle cell anemia, sickle cell disease, is one of the most famous, where thanks to the fact that we're born with redundant hemoglobin genes, we have a set of fetal hemoglobin genes that produce our hemoglobin when we were in utero. And then we have a set of adult hemoglobin genes that around the time of birth begin to take over for our fetal hemoglobin genes. So those of us who have mutations in the adult hemoglobin genes that result in sickle cell anemia can be rescued if only we can figure out a way to turn back on the fetal hemoglobin genes that are normally silenced around the time of birth. So that's what CRISPR Therapeutics and Vertex and others have had great success doing. That's an example where you can mess up a gene, and they typically target a gene that's responsible for keeping the fetal hemoglobin genes turned off. And if you mess up that gene in that case, you can benefit a patient by turning on the redundant fetal hemoglobin genes instead to compensate for their mutated adult hemoglobin. But that's a unusual case, a pretty fortunate and ideal setup where messing up a gene can actually restore the function of a healthy gene that was otherwise turned off. And unfortunately, in genetic diseases, there aren't that many cases where such a situation exists.
And so how does prime editing factor into all this? Do you see some major advantages or challenges that prime editing will have in comparison to base and CRISPR-Cas9?
Yeah, so base editing currently can make mostly 4 changes. It can change C's to T's, T's to C's, A's to G's, or G's to A's. Those 4 kinds of changes are collectively called the transition point mutations. And fortunately, the transition point mutations are the most common kind of single letter mistakes that we observe in disease and in evolution in general. And single letter mistakes are the most common class of mistakes that are associated with genetic disease. So, for example, in our recent progeria study, we corrected using a base editor, we converted the mutation, which is a C to T mutation that causes most cases of progeria. We converted that T back into a C by converting the A on the opposite strand back into a G. And as a result, we could rescue many of the symptoms of progeria in mice that were engineered to have the human progeria gene. But there are many kinds of genetic changes beyond those 4 C to T, T to C, A to G, G to A changes. For example, sometimes an A is mutated to a C, and that causes a disease. Sometimes it's not a single letter misspelling, but instead the insertion of 4 bases. If you insert TATC at a certain site in a gene called HexA, you end up with Tay-Sachs disease. If you delete 3 nucleotides in a certain location in the CFTR gene, you end up with cystic fibrosis. So those are examples of mutations that can't be fixed directly with base editors. And so we developed prime editing as yet another gene editing technology. Prime editing enables all 12 possible base-to-base changes, as well as small insertions and small deletions, to take place in a programmed way in human cells. And in unpublished results, we've also had some success in animals. And a number of labs have now published preprints or papers showing the use of prime editing in a wide variety of contexts, ranging from mouse embryos to plants to even adult mice.
Incredible. And, I think maybe it would be helpful if you just discussed how involved you were with base and prime editing, because maybe you're being a little bit too humble on when you're discussing these topics, but it'd be really helpful to hear, how this all sort of came about and your involvement with it.
Yeah, all research projects are team efforts in modern science, especially the life sciences, where experiments are difficult, expensive, and take a long time. So, I really give enormous credit to a very talented team of graduate students and postdocs who led all of those projects. Alexis Comore, former postdoc in the lab, who's now a professor at UCSD. When she was an incoming postdoc in the lab, started iterating project ideas with me, as I often do for incoming postdocs. And she had initially proposed working with systems that might modify RNA in a sequence programmed way. And the brainstorming that we had, the email dialogue that we had, stimulated some thinking about what seemed to be increasingly a major problem facing the gene editing field. Namely, gene editing was getting very good at disrupting genes by cutting them. But when I looked at many, many genetic diseases, the answer was clearly, well, it's not obvious how you would mess up the gene to benefit the patient. We need to precisely change one base pair to a different base pair in order to fix the disease. Or perhaps we need to install that mutation in order to study the disease and ascertain the relationship between the mutation and the disease. So all of those considerations led us to focus instead on how important it might be, how impactful it might be to change a single base pair to a different base pair without cutting the DNA double helix. And that was really the start of base editing. And unlike most projects, the very first base editor, the cytosine base editor, actually unfolded pretty much like we outlined it in those emails back in 2013. We imagined taking advantage of the fact that CRISPR-Cas9 can unwind DNA, because the enzymes that do most of these chemical reactions on the bases, at the time, all of them that were known to convert C into something that looks like T required single-stranded DNA. Since then, Joseph Mogus, the University of Washington, discovered one that works on double-stranded DNA, which we turned into the first mitochondrial base editor, as a side story. But the recognition that CRISPR-Cas9 was well suited to unwind DNA in a way that would facilitate doing a chemical reaction on an individual DNA base was really the start of Alexis's postdoc project. And so once she arrived, she worked for a couple of years and pretty quickly got the system to work. And then the adenine base editor, which is especially important because it turns out that the conversion of A's to G's and T's to C's would correct about half of known pathogenic single letter mistakes. That was led by former postdoc Nicole Godelli, who's now at Beam Therapeutics. Nicole bravely took on this project of trying to basically develop a new class, a second class of base editor, that instead of converting C's to T's and G's to A's, would do the opposite, would convert A's to G's and T's to C's. And the challenge with her project is, unlike the cytosine base editor, where we could borrow an enzyme from nature that we already knew would convert C into a base that looked like T, there was no such enzyme that would convert A into a base that looks like G in DNA. And so Nicole bravely agreed to break one of the rules in the lab, which is you don't start a project if step one of the project is we have to evolve a whole protein just to get started. And she agreed to try to evolve the first deoxyadenosine deaminase. That is an enzyme that would convert A into a base that looked like G in DNA instead of in RNA or in free floating A. And she succeeded in doing that. So her, step one of her project was literally evolve this new enzyme that hadn't existed before, to our knowledge. And she did, and then she beautifully integrated it through a lot of fits and starts and a lot of careful analysis into a mammalian cell base editing architecture. And then she also succeeded in developing this engineered molecular machine that now can find a target base pair, an AT base pair, and convert it to a GC base pair quite cleanly in human cells at positions that we specify. And that adenine base editor is now the basis of a number of both therapeutic and research programs in industry and in academia. And then with prime editors, had the benefit of another former postdoc in the lab, Andrew Anzalone. He's now at Prime Medicine. You can see this trend where many of the postdocs in my lab that I always thought from the beginning, based on their interests, would end up in academia, instead realized the excitement and the potential of science that they developed in the lab, and instead decide to be one of the early employees in a company founded to bring that science to patients. And so Andrew Anzalone interviewed as a prospective postdoc. He was an MD PhD student, did research in Virginia Cornish's lab at Columbia. And in his interview with my, rather discerning group, shall we say, they normally really kick the tires on postdoc candidates. I encouraged Andrew to present an idea, which he talked to me in our one-on-one meeting just 30 minutes before his group meeting presentation. And his idea was basically the essence of prime editing, was his thought that perhaps you could take a freshly nicked piece of DNA, grab that nicked 3' end, and use it as a substrate for reverse transcription. In other words, directly copy from RNA onto DNA the bases that you wanted to add to that target DNA strand, all in a living human cell. At the time, it sounded pretty far-fetched, but none of the steps sounded crazy. Just the project by itself would require 4 or 5 almost crazy things to go right. So there was a lot of skepticism, but I thought, I thought that the impact could be high enough that it was worth taking the risk. So I made him an offer. I was fortunate that he came, and then Andrew, I think this will be in the lore of our lab forever. He got the project to work in a frighteningly short period of time for something that looked on paper to be one of the most difficult projects in the lab. So from the time he arrived to the time we submitted the paper was only 1 year and I think 7 or 8 days. And so, I made this sort of joke during the Cold Spring Harbor meeting in which I gave the very first presentation to the world about prime editing. At the end, I just was acknowledging everybody and sort of ad-libbed once I got to Andrew that I was looking forward to seeing what he'll do in the second year of his postdoc. So Andrew was totally amazing. And, all of these 3 projects, even though I'm highlighting the people who led those projects, they all ended up with half a dozen to a dozen people from the lab contributing. And that's really been one of the most important aspects of our lab, is that people are largely chosen and are encouraged to develop on the basis of how well they work with others. And people really work not only very hard, but they work to try to maximize everyone else's success, not just their own.
Yes, and strong leadership also helps. But, it's interesting you brought up the companies and, leveraging that expertise to come to the companies, which is really interesting. Also, the structure in general of your companies and how many you're involved in is pretty spectacular. You must be very busy. So, an interesting example is that Beam has a stake in Prime. So John Evans, Beam CEO, of course, was the interim CEO for Prime, and they can really use each other's and leverage each other's technologies and expertise. So I'm curious to know, as we see the field kind of evolving, there seems to be different camps in terms of how each company will interact together, especially with one founder that they all have in common. So could you ever see yourself maybe doing sort of an umbrella company with a few subsidiaries that can all, share their IP and expertise? Or do you think, as you continue to build up your company portfolio, they would all be sort of separate entities?
Since I don't provide the financing for these companies, my own plans for how they're set up may not be as relevant as your question seems to imply. But in the case of Beam and Prime, it was a really interesting situation because once Andrew started getting results in mammalian cells, I think everybody in the lab knew that something special was happening. And one of my fears, frankly, one of the downsides of that excitement was I remember in the summer of 2019 spending a lot of sleepless nights wondering what the impact would be on Beam, on the patients that Beam was very, very well poised to help. Beam was and continues to do great science. They have terrific people, including a number from my lab. And, knowing exactly kind of the considerations you described, I started to think about what would the best way be to bring prime editing to patients in a way that was synergistic with rather than competitive with base editing and Beam. I think the first decision I made is, well, if we end up creating a prime editing company, we should do so by involving the same board members, basically, of Beam, so that we can make sure that the patients that are already poised to benefit from base editing wouldn't be impeded in any way, wouldn't have the progress on the development of their therapeutics slowed down or distracted in any way by prime editing, but instead there could only be benefit. And so, with the help of the Beam investors, the Beam board, Arch, F Prime, GV, we set up Prime Medicine to really be sort of a sister company, where, as you point out, Prime actually had interim management from Beam. I think the leaders at Beam have been fantastic, incredibly respectful of the science, but also very skillful on the business side. And as a result, from the get-go, a relationship was set up where Prime and Beam would benefit each other. And the nature of their agreement, which I think people can read, it's public, or at least aspects of it are public, is really designed to maximize the chance that patients benefit from both base editing and prime editing, and that the companies don't expend resources either fighting each other or wasting resources doing redundant things, knowing that in the end, only one therapeutic for a particular disease might be the best, or even worse, simply spending hundreds of millions of dollars to learn the same delivery or manufacturing expertise that is such an important and sometimes overlooked aspect of the gene editing industry. So in the end, I'm pretty happy with how both of those companies ended up being set up, despite the sleepless nights that, ironically, that this success, this excitement of realizing that prime editing was working in mammalian cells caused, because I immediately started to wonder, how can this best be brought to patients in a way that helps rather than distracts from the great work Beam was doing?
Yeah, I think you sort of, approach that really, really well. I love the ideas of the companies working together to find, cures or potential cures for really hard to treat and diseases and unmet needs. So I think them working together and doing what's best for patients is so important, as you just outlined really well. Here we're talking a lot about humans, but this can also be used in agriculture. So just curious, I haven't seen anything in terms of pipeline, if that's something that you're interested in or, going after in terms of the companies or just personally, but even when you mentioned, the sky in your room there, it's something that obviously seems very passionate to you. So I'm just curious if that's something you're thinking about either from a company perspective or a side hobby.
Yes. So with the caveat that I know almost no plant biology, at least by plant biologist standards, I think genome editing in agriculture is poised to have a major impact. Along with Fun John and Keith John, I co-founded Pairwise Plants, which is a genome editing ag company. It's based in North Carolina, and they are using these new technologies, including base editing, to create more nutritious or more hardy crops. And I think they have some exciting developments. I'm not sure what's been publicly announced, so I'll refrain from going into details, but I think it's easy to anticipate that genome edited crops, may actually be the first accessible product of gene editing for much of society faster than genome editing therapeutics, because the timelines are a little bit different, even though it's not trivial to bring a new crop to market, for sure.
Right. That's fascinating. I actually did not know about that company, so thank you. That's really, really interesting. So to pivot a little bit, talking just about, I guess, thinking about how and when costs may decline, obviously in the context that if we're providing, life-saving drugs, then the idea is that those drugs will be more expensive, which makes sense, per life year gain that someone will get. And also, for the lack of need to continuously go in for treatment. But if you think about costs declining eventually, which will likely happen, at ARC, we typically think about it from a rights law perspective, which is just a framework to forecast cost declines. And obviously, without using any specific calculations, not putting you on the spot, but, how quickly do you think that costs are really going to decline for gene therapy or gene editing? And do you think this will happen in a way that therapies can become more democratized?
Yeah, I sure hope so. And I think everybody in the field is hoping and anticipating that that will be the case. Virtually every key component of gene editing, from discovering and developing the editors to improving them to levels of therapeutic editing efficiency and specificity that begin to be therapeutically interesting, to delivery into the right types of cells in ways that ideally don't cause undesired side effects, to manufacturing. All of those components are in some ways frontiers in when applied to gene editing. I mean, macromolecular delivery is an old, venerable field, but delivery of gene editing agents it has almost stimulated this renaissance of the field that has inspired all sorts of crazy new ideas, some of which actually work pretty well. So I think there will be a rights law decrease in cost, as well as probably a Moore's law decrease in cost, because the speed with which people are joining and advancing the gene editing field continues to be breathtaking. I mean, many times in the life sciences, or in the physical sciences for that matter, there's a hot discovery that inspires several years of students and postdocs to join that field. I don't think I've been a part of one that has had such a sustained gravitational pull as the gene editing field, where now we're probably a whole scientific generation has passed, i.e., going from a graduate student to being a professor. And yet the desire is as strong as ever, the interest is as strong as ever to continue to advance that field. So the rate of progress is stunning, shows no sign of letting up. And I think all of us would be incredibly disappointed, and frankly would I think refocus our efforts to make sure this doesn't happen if it started to look like any of the therapeutics that might come from the gene editing revolution would only be able to serve a small number of incredibly wealthy patients. That would be, I think, a devastatingly bad outcome.
Yeah.
So I'm optimistic that because of Wright's law, Moore's law, and the ingenuity and the hard work of everybody in academia and in industry that's working in this space, that we will see the second and the third and the fourth gene editing therapeutics, whether it's from CRISPR nucleases or from zinc finger nucleases for that matter, or from base editors or from prime editors or from mitochondrial tail-based base editors. I think we'll see the nth example of those come at much reduced cost to development, and therefore cost to the patient than the initial examples.
Yeah. Gene editing and gene therapy, it's so exciting. We did some research where we looked through clinicaltrials.gov. We noticed that gene therapy and editing trials have increased about fivefold since 2010. So that, based on our estimation, would be roughly 170 gene therapies that are likely to be approved and commercialized during the next decade. Obviously, a massive increase to what was previously done. So, I'm curious to know if you think that gene therapy and gene editing could be a sort of a dominant form of therapy, and if they could ever kind of replace the small molecules or biologics of the world.
In some cases, I would agree with that. Of course, it's important to appreciate that for the most part, gene therapies and gene editing therapeutics are only poised to benefit conditions that have a strong genetic origin. And, there are many conditions. If you think of all the drugs you've taken in the past month, not insinuating anything, but, there are probably things like you took Advil, which has nothing to do directly with a genetic mutation, probably. So I think there will always be a need for small molecule therapeutics that can uniquely address some conditions, some diseases that may not be appropriate or may not be possible to address with gene editing or gene therapy. Here I'm using gene therapy to refer to gene augmentation therapy, where you're providing extra copies of a healthy gene. But I agree with your overall assessment that it, I think, would shock nobody if a substantial fraction of new drug approvals in the coming decades were gene editing and gene therapy drugs, since they represent such a large fraction of investment and of activity in the life sciences right now. And of course, the need is urgent, because for the vast majority of the initial indications that companies are pursuing in these areas, there is no effective treatment known.
So thinking about your early work, I only know it from the amazing papers you publish, but thinking about, I've seen papers from Science and Nature, and discussing, using DNA for chemical synthesis. I haven't seen too much follow-up on this. I'm just curious, have you moved more towards just focusing on gene editing, or are we going to see more about DNA for chemical synthesis in the future?
Yeah, so we continue to do DNA templated synthesis to use it to discover bioactive small molecules, some of which have relevance at least as probes, if not as therapeutic leads. And more recently, we're using that platform to evolve sequence-defined synthetic polymers that have really interesting binding or even catalytic properties. So evolving non-natural catalysts that, just like proteins, can fold into specific three-dimensional shapes that catalyze chemical reactions, but are not made of proteins and have side chains that we entirely specify with a genetic code that we entirely specify. That's one of the project areas that we're working on in that side. So the lab continues to have three subgroups, of which one is DNA templated synthesis, DNA encoded chemistry of both small molecules and synthetic polymers. The second of which is protein evolution and protein delivery, and the third of which is gene editing. It's just that, as is often the case in the life sciences these days, gene editing seems to have an unusual gravitational pull. But I've seen this before, so I've learned to allow students to vote with their feet, to follow their interests. But there have been times in which our DNA templated chemistry subgroup was by far the largest of the three. There have also been times in which our protein evolution subgroup was by far the largest of the three. So I think one of the great benefits and privileges of being an academic is you get the opportunity to work with these incredibly talented students who stay the same average age every year, even as I get older and more senile. And you have the freedom to really allow them and yourself to follow your curiosity. I think DNA encoded chemistry and synthetic small molecule discovery, synthetic polymer discovery will always be super interesting and potentially quite impactful. But you're right that right now the plurality, not the majority, but the plurality of students and postdocs in the group are interested in gene editing in the lab. That said, other subgroups within our lab have really benefited the gene editing subgroup. So our protein delivery and protein evolution subgroups and the techniques that they've developed have been repeatedly used by the gene editing folks. So all of our current base editors, Cas proteins, and even some of our new unpublished prime editors have been evolved in the laboratory and are now quite different than the versions we initially reported. So there's a lot of synergy between different areas that will keep emphasizing to me the importance of maintaining, a diverse research portfolio and not simply researching only in one narrow slice of gene editing, for example.
Yeah, and you mentioned, Andrew went from his project basically took a year to the first paper, which is so exceptionally fast. So definitely a lot of interesting stuff coming out of your lab and interesting students with really good leadership, obviously. I was really excited about the timing of this podcast. One of the reasons was because I've been reading all about the next frontier, which we keep hearing about, which could be proteome editing. And it this is like hot off the press. I read in the Harvard Gazette, and then I think there was an article in Science last week that you can use Botox, and that you and your team can use this toxin to target a number of different proteins, and that it could really help with a few things, which I found really fascinating, being regenerative medicine with neuroregeneration, which, as we know, is one of the most difficult things to target. Really difficult to find solutions for it. And it can also help with inflammation, overreactivation of an overreaction from the immune system, like cytokine storm, which a lot of people probably have heard of from COVID-19. And I loved in the article, it said something like Botox, you either know it because it gets rid of your forehead wrinkles or because you think of it as a poisonous chemical, which I was like, yeah, that's, I guess, what people...
Yeah, the miracle poison, it's been called. Yeah, that was a really cool project that really grew from the contributions of many people. Michael Packer was a graduate student in the lab who is now at Beam Therapeutics, actually. But Michael worked on PACE projects, and one of his major projects was trying to use a PACE system. Sorry, PACE is phage-assisted continuous evolution. So it's our very rapid laboratory evolution system that we now apply to lots of proteins, including genome editing proteins, base editors, CRISPR-Cas9, prime editors. So PACE allows a protein to evolve at a speed that is about 100 times faster than we could previously evolve it. The secret sauce behind PACE is that it takes place continuously because we've mapped every step of the Darwinian evolution cycle onto the life cycle of a bacterial phage, a bacterial virus called a filamentous bacteriophage. And therefore, we can complete an entire generation of Darwinian selection, replication, mutation. We can complete that cycle in sort of minutes to hours instead of the usual week or so that it takes to do manual traditional protein evolution. And that means we can evolve proteins through hundreds or even thousands of generations of Darwinian evolution, which we could never do before. If you evolve a protein through 2,000 generations, which has been done in the paper you just read, normally it would take you 2,000 weeks, which if you do the math is not really compatible with a PhD or postdoc timeframe. So using this PACE system, two former group members, Brian Dickinson, who's now a professor at the University of Chicago, who first developed the selection, and then Michael Packer, now at Beam Therapeutics, showed that you could take a protein and dramatically modify its specificity. So instead of cutting its native target, it still cut that target, but it would also cut some very different target. So we took TEV protease, which is just a sort of famous protease that recognizes 7 amino acids, and we used this PACE system to evolve it to cut IL-23, human IL-23, which differs at 6 out of 7 positions recognized. But we realized that those proteases would still cut the native substrate of TEV protease. In other words, we had expanded their protease specificity rather than really reprogramming it from one protein to a different protein. And so then came current postdoc in the lab, Travis Blum. So Travis developed a negative selection system working with Michael shortly before Michael left for Beam. And then Travis repeatedly refined and applied that new positive and negative protease selection system, which can now select for the ability to cleave a desired protein and against the ability to cleave one or more off-target proteins. And he applied it over and over again successfully to, I think, brilliantly chosen class of proteases brought to our attention by Ipsen pharmaceuticals scientists called botulinum neurotoxin proteases. So Botox, of course, most people know is the thing that you get an injection of to make your wrinkles go away. It's actually used as a therapeutic to treat a variety of conditions. But it's remarkable because Botox has a light chain and a heavy chain. They come together to form the toxin protein. As everybody probably knows, botulinum neurotoxin is very, very potent. So a typical dose might be a nanogram, which is kind of stunning to imagine. The light chain does the protein cleavage, that's the protease. The heavy chain self-delivers into neurons. And researchers have begun to reprogram that heavy chain to also hit cell types that are different than the cells normally targeted by botulinum neurotoxins. But proteases, protein cleaving enzymes that are selective for a specific sequence, in principle have enormous potential as biological tools and as even human therapeutics, except there is no immune system for proteases. Unlike antibodies, where we can generate an antibody to just about any kind of antigen using an animal immune system or various laboratory mimics of those immune systems, such a system hasn't really existed for proteases that allows us to take a protease and reprogram it to cut a very different protein selectively without causing it to become more promiscuous. So Travis used this continuous evolution system with a positive and negative selection built in and evolved not one or two, but 4 different reprogrammed botulinum neurotoxin proteases that now hit their new target and don't hit their original target anymore. And in 2 of those cases, there was no known botulinum neurotoxin protease that could cleave the new target. So it was really an ambitious shot in the dark that the system was powerful enough to be able to cleave a protein called PTEN, which has no obvious sequence similarity to substrates that are natively cleaved by these botulinum neurotoxin proteases. And Travis succeeded in evolving a pretty reasonable protease that will cut PTEN, but doesn't cut any of the normal substrates cleaved by botulinum neurotoxin proteases. So, this phrase proteome editing is really an acknowledgement that the era of modern gene editing began when researchers figured out how to program nucleases, DNA cutting enzymes, to cut DNA sequences of our choosing rather than those specified by natural enzymes. And so while it's quite different and admittedly, it's really an apples and oranges analogy, but the idea of being able to now program proteases to selectively cleave proteins of our choosing rather than the protease substrates that are normally chosen by naturally occurring enzymes raises all sorts of interesting possibilities, since virtually any disease and really any biological process you can think of probably has at some critical point a protein where if you could selectively cut that protein or activate it, something you can do with a protease, or change where it is in the cell, or change its lifetime, change its post-translational modification state, that you could imagine at least a way where that protein cleaving event could interfere with, modulate that biological process in a way that might benefit a patient. So, it's in very early days. We've repeatedly demonstrated that this platform can reprogram botulinum neurotoxin proteases to be selective, yet to cut new proteins. But what we do with it, I mean the scientific community, not just our lab, remains to be seen, and is, I think, one of the more exciting aspects of thinking about it in terms of what if we could actually cut some significant fraction of different proteins in the human proteome. In some respects, it's actually an easier problem than gene editing because there are fewer protein sequences than there are DNA sequences. And the math of specificity of recognizing a specific protein in the human proteome, I think, tends to be a little bit more favorable than the math of recognizing a single site in the human genome. But it's part of why I love being a professor. There's all sorts of crazy ideas now we can imagine doing with this system, which, at the time that Travis was in the thick of it, trying to refine the system and then use it over and over again to make sure that it wasn't just a fluke that his first or his second or even his third success worked, was probably a lot of hard work. I mean, it was a lot of hard work. And now to be able to have fun with it and imagine what we might be able to do with it and what other labs might be able to do with the system is really exciting.
So you're a little bit busy between base editing, prime editing, now proteome editing. It's really inspiring. So in addition to being, in my mind, one of the most prolific scientists, definitely of our time, maybe forever, you also have co-founded Editas Medicine, Beam Therapeutics, Prime Medicine. How do you divvy up your time and sort of how involved are you in the companies in the day-to-day?
Yeah, so I'm not an employee of the companies, of course, and I am careful to maintain a necessary firewall between what my lab does and what the companies do, as all my institutions require, and as is common sense. But I care deeply about making our science transition from the pages of a journal to some kind of societal benefit, whether it's more nutritious crops or new medicines for diseases that can't be treated otherwise. So I take super seriously my responsibilities for each of these companies to try to do what I can, along with my co-founders, and of course, most importantly, the leadership and the incredibly hardworking scientists in each of these companies, to try to uh, make sure that the company succeeds or fails, not because we didn't try hard enough or didn't put enough money in or couldn't recruit the best people, but if it does fail, it should only fail because in the end we weren't smart enough to figure out some important problem. But I think history suggests that if you put science that's reasonably solid together with strong support and really great people and great leaders, like some of the ones you've already mentioned, like John Evans, really good things happen. So it's been humbling actually to see how quickly and how successfully each of these companies has transitioned technology from our lab and from other co-founders' labs, and not only applied it, but in many cases taken it in a slightly different direction or improved it in a unique way, all aimed at doing whatever it takes to bring safe and transformative, efficacious new therapies to patients who are suffering from conditions that really don't have much hope right now.
So I know you have to wrap up and go in just a few minutes here, but speaking of John Evans, when I asked him, I'm really excited. We're going to be talking to David Liu. What should I ask him that maybe people don't know? He said, well, ask him about his blackjack card counting. So would love to hear more about that.
That might take a whole nother podcast, but as a teaser preview, for about 4.5 years or so, I taught an informal class that met every week in my office. Attendance was 100%, pretty much, and the class was on developing creative new mathematics systems that could beat blackjack. And we actually went to casinos all across the country and practiced tested our systems with quite successful results. And it was a great experience for me, mostly because I got to know a bunch of remarkably talented students in a very different way. And it was also the smallest class I've ever taught. Before that, the smallest class I taught was about 60 students. So this was a much more intimate kind of classroom and getting to know students experience. And of course, it was incredibly fun, and all sorts of hilarious things happened, as you might expect, because while it's not illegal, card counting is not appreciated by casinos. And so there was definitely a social aspect, a social engineering aspect to the experience that was just as important as the math and theory aspect.
Yeah, and I think when we had talked on a previous call, you had mentioned to get into your lab, you can interview for the blackjack portion of the lab or the scientific portion of the lab, and only one student may have gotten into both. I think you had mentioned.
And he chose the blackjack team. So yes, there's a rule when I had the blackjack team that you could either try out for the lab or try out for the team. Well, you could try out for both, but you couldn't do both. And there was one incredibly talented graduate student, David Cox, who ended up joining Fun John's lab, who is the only person who received an invitation to join both. and he chose the blackjack team.
That's amazing. I know
And then he did wonderful things in Fun John's lab.
Amazing. I know you need to wrap up. Last question. What is going to happen in 5 to 10 years? What's your prediction? Where are we headed in terms of gene editing? Or is there something you can imagine beyond gene editing in 10 years that you'll be working on?
It's tough to predict. I think, 5 years ago, if you had told me that we would be using engineered laboratory machines to change a single base pair in an animal that suffers from an otherwise incurable genetic disease and fix aspects of the disease, I would have said that sounds a little bit crazy. But I would be surprised and disappointed if within 5 to 10 years there weren't multiple gene editing therapeutics. They will inspire all sorts of additional ways of manipulating the molecules of life that currently seem like science fiction, the way base editing and prime editing seemed like science fiction 5 years ago. We should all feel very fortunate to be alive during this time when we're witnessing one of the great revolutions in the sciences unfold before our eyes.
Completely agree. Thank you so much for all the insights you provided today. This has really been a wonderful podcast and just a really cool experience for me to be able to chat to you about all this stuff.
Well, thanks for your interest and for the kind invitation to come chat. I really enjoyed it.
Great. Thank you so much.
Okay, take care, Allie. Bye-bye.
Bye. Enjoy the lecture.
Yes, I got to go teach.
ARC believes that the information presented is accurate and was obtained from sources that ARC believes to be reliable. However, ARC does not guarantee the accuracy or completeness of any information, and such information may be subject to change without notice from ARC. Historical results are not indications of future results. Certain of the statements contained in this podcast may be statements of future expectations and other forward-looking statements that are based on ARC's current views and assumptions, and involve known and unknown risks and uncertainties that could cause actual results, performance, or events to differ materially from those expressed or implied in such statements.